This is the second section of the fourth chapter on agonist deficiency, in which we are concerned with the nature of the isomerization, the gating isomerization between C and O. So in the first part of chapter 4, we defined the parameter phi, showed its distribution, and described how we measure it. And that is shown by the picture here in the bottom right, which is phi is the ratio between these two blue lines, the effect of a mutation on the transition state energy relative to that on the open state energy. In this chapter, we are going to interpret the value of phi, and particularly the distribution of phi. And what we found was that there are 5 discrete populations of phi that range from 0.95, which is that the transition state energy change was almost the same as the open state, all the way down to close to 0. So sometimes in some residues, the transition state energy is O-like, that means high phi, and some it's C-like, that means low phi. And that's the purpose of this chapter, this section of chapter 4, just to interpret that. So here are those 5 populations of phi value, and what we think that phi is going to tell us is the number of intermediate states between C and O, which is one less than the number of intermediate rearrangements or transitions between C and O that link C and O, and their order of appearance at the transition state, which I will call the sequence. So the model that we use is shown by this energy landscape. That is that there are short-lived intermediate states that separate the stable end states A2C and A2O. And if we say there are 5 transitions, that means there are 1, 2, 3, 4 intermediate states. So what I'm about to describe to you is simulations that are described in this 2005 paper. So if you perturb the first step in the process, which is shown here by these purple lines, you would either make that first step more favorable or less favorable, and that results in a larger and smaller equilibrium constant. You can measure the rate constant and what was the effect of the perturbation on the rate constant. And when you perturb the first step, you find that the effect on the rate constant was almost the same as in the equilibrium constant. But if you perturb further and further into the reaction change, this is the second, gives you a lower phi value, the third gives you a lower phi, yet lower, fourth gives you yet lower, and at the end you get even lower. So as you perturb deeper and deeper into the reaction chain, you get a smaller and smaller phi value, and that is totally intuitive, because you can imagine if you perturb in the beginning, you're going to change the rate constant much more than if you perturb at the end by the same amount. And if you perturb the last one, you're really not going to change progress through this, the stochastic progress through this energy landscape. So the fact that there are 4 phi populations, 5 phi populations means that there are 4 intermediate states, and we can put them in sequence from purple being the earliest and brown being the latest. So we can go from early to late in the reaction chain, and we now know there are 4 intermediate states and 5 intermediate transitions. And although this was done by simulations in 2005, we soon thereafter figured out the analytical form for calculating phi. The key assumption here to do that was to make the lifetimes of these intermediate states vanishingly small relative to the end state lifetimes. The third thing we learn from phi value analysis is the actual landscape for the energy barrier region and the position of the transition state. So again, in those analytical papers shown down below, we were able to calculate the roof of the barrier region and isolate within there a point called the transition state, or the committer, which is the point at which if you drop a ball, it would rapidly end up at C or at O with equal probabilities. And there's thermal energy causing it to move across this landscape. So from the distribution of phi values, we can calculate an actual landscape for the transition state ensemble and the actual position of the committer within that ensemble. And that means we can generate a reaction scheme which is defined by the number K, which is determines the depths of these wells. So under the condition that these wells all have the same depth, if we use a K value of about 350,000 per second, that gives us these following distributions that we would record in the patch clamp had we a patch clamp that could go down into the microsecond range. That is, we would see a single open component, but we would see 1, 2, 3, 4 different shut components that correspond to, plus the 5 different shut components. The longest one corresponds to the C state, all the way in C, and these components correspond to some population along the transition state. So the slowest of these transition state components is about 6 microseconds, which is the same as the lifetime of flip and prime. Indeed, we used the flip and prime lifetimes to come up with a 350,000 number here. And we predict that there are shorter components, but the resolution of the patch clamp is roughly around 10 microseconds here. So what you would only see is this rising phase of the aggregate, which is the dark line here, of the sum of all the components, which you would obviously only fit to a single component and call it 5.6 microseconds. So that's why we think flip is really an aggregate measure. It measures a bunch of different states. So we learn the landscape and position of the transition state, and so we can add that to, and these intermediate states are on the order of microseconds or hundreds of nanoseconds. We also learned something else that's actually quite important, and that is we can pinpoint the energy change caused locate them in the structures of the protein. And we can do that because of this data shown in this panel over here, which are mutant cycle analyses. What a mutant cycle analysis is, you make a mutation to residue A and measure the effect on the gating equilibrium constant. You make a mutation to residue B and measure the effect on the gating equilibrium constant. And then you make the pair of mutations and measure the effect on the gating equilibrium constant. And if you compare the pair effect to the sum of the individual effects, and the difference between those is a interaction energy. So if the pair and individuals, some of the individuals were identical, that would give you an interaction energy of 0. And you either get favorable or unfavorable interactions. And we plot those interaction energies on the y-axis versus the separation of the two amino acids, the C-alpha distances. And we're using GluCl as a template structure because the time we did this, there was no good structure of the acetylcholine receptor. So I want to point out there are noises here. There are noises from measuring these interaction energies, and there are noises in our distances because of the difference in protein and the fact that we're using C-alphas. But in any case, the pattern is pretty clear that if we assume that our noise level on the y-axis is 2 kT, or 1.2 kcals per mole, that's given by this orange bar. It's probably a little less than that. It may be more like 1 kT, which is this, or these dashed lines. But what we see is that for the most part, there is very little interaction between the residues, that the sum and the pair are approximately the same. There are the ones that do deviate from that are all close together. This line is about between 15 and 20 angstroms. Somewhere in that range, is where we get the deviations, even though within that range there are many, many pairs that do not interact. But there are a few that do interact positively and negatively inside of there. There's also a couple here that I will mention later on that are further away that do interact. These are interesting, but we don't really understand them, so I'm not going to make a big deal out of them. But what we learn out of this is that phi values can be mapped onto structures. That is, because the energy deposition is exchanged with the environment is local, that we can pinpoint position in the structure of the protein the energy change and make a map of energy changes, and specifically phi values is what we're going to be interested in. The lack of energy coupling allows us to make a map of phi, and that is shown here. These are the amino acids colored according to their phi value, mapped onto the structure of GluCl, with the arrows at the left marking either the neurotransmitter binding sites up above or the extracellular transmembrane domain interface down below. The white balls represent amino acids that we made mutations of, but the change in the log equilibrium constant was too small to allow a slope to be estimated from the reefer. So we do not know what the phi values of those residues are. These residues were mostly superficial. So we can take this map and explode it according to phi value, and you can see that the high phi residues are mostly at the binding site. The blue 0.8 phi residues are mostly in the extracellular domain. The green residues at 0.6 are concentrated in the transmembrane domain, and the lower phi residues of 0.3 are also in the transmembrane domain, but concentrated at the gate, but scattered around a bit. There are some exceptions to this. You can see that in the extracellular domain, in the high phi residues are not only at the binding sites, but there are also a group in the M2M3 linker at the interface, and it turns out that these are the ones that are coupled energetically. There are also some low phi residues at this interface as well as down below. They're not only at the gate region. And there are complications to measuring phi. For example, these guys at these high phi residues at the interface are mostly loss of function residues, and it's possible that a Hammond curvature in the reefer is affecting the phi values that we get from those. That needs to be investigated further. And also there are, as I will describe later on, other secondary perturbations that might change the phi value that you get here. For example, these guys here also at the interface. So I'll talk about that in a moment. But in general, there is this clear spatial localization of phi values. Although most of our measurements were phi values of amino acids in the alpha subunit, in some regions of the protein we managed to measure phi for all 4 subunits. And this shows the map of phi on the M2 helix. This is the helix that lines the pore in the transmembrane domain. And the gate region is between residues 13' and 9' at the equator of the M2 regions. And you can see that the brown residues, the phi equals 0 residues, are mostly at the gate region or below, and in non-alpha subunits, although this 9' residue in the alpha subunit was also phi of 0. This tail here represents the M2M3 linker, and these are those high phi residues that are only present in the alpha subunit, the ones that are coupled to the binding site. So the brown residues are mostly at the gate region, and you can see that in general that the alpha subunit has higher phi values than the epsilon subunit, which is higher than also the delta and the beta subunits. There's a lot more white residues in these non-alpha subunits than there is in the alpha subunit. Those are the ones that don't change the energy of the gating equilibrium constant very much. So what we learned from the distribution of phi values shown here in the upper left is the number of intermediate transitions between C and O. There are 5, separating 4 intermediate states. Their sequence from purple, blue, green, red, and brown, going from closed to open. The energy landscape, and particularly the location of the transition state or the committer, shown here by the double dagger, and we can make a map. So we know the first event we have called hold. We discussed this in the first three chapters on agonist efficiency. This is the transition of the binding site from between low and high affinities. But now I'm going to give names to each of the other ones based on the structural changes that have been observed between closed and open in each of the domains shown to the right. So in the extracellular domain, shown to undergo a small twisting motion, as well as an unblooming motion or a contraction as from C to O. But I'm going to call that one, give that the name twist for the conformational change, the blue conformational change. In the transmembrane domain, the membrane helices the rearrange. So I'm going to call this one tilt. And in at the gate region, there is a pulling apart of the M2 region in the vicinity of the gate. And finally, I'm going to associate the lowest phi with the fusion of the water compartments that takes place to initiate ion conduction. So the 5 intermediate transitions I'm going to name hold, twist, tilt, rip, and pop, with hold being the affinity change and pop being the conductance change, and the twist, tilt, and rip being the intermediate transitions of the extracellular domain, transmembrane domain, and the gate region. And now we just put these in some animation to show that this is their sequence of conformational change. Pop I'm not showing because that's in the other non-alpha subunits as well. So what we learned from this phi analysis is the nature of the dynamics of chemodynamics, the gating conformational cascade, which connects conformational changes at the binding site that are initiated by a change in affinity for the agonist, that it pass down the long axis of the protein until they reach the gate, and finally allow the water compartments to fuse. This is a close-up of a discrete boundary between the 2 phi blocks, one between the blue group of residues at 0.8 phi value and the green group of residues at 0.6. That is between the extracellular domain and the transmembrane domain. And I'm showing you a very discrete boundary in the alpha 1 helix at a pi helix in the alpha M1 transmembrane helix. So that's shown here by this arrow here. These 2 blue beta strands are beta strands 9 and 10, with the neurotransmitter binding site being up above. There is a linker that joins these with the M1 helix, and the brown residues here on the left represent the gate region. And I'm showing on the right is a blow up of this arrow, the region near the arrow. At this region, there is a pi helix caused by this proline, a very conserved proline in M1 that interrupts the hydrogen bond network. And there's a pi helical segment here. And it turns out that there's a very discrete boundary between residues, that a residue here, 217, which has a phi value of 0.84, and the next residue, 218, has a phi value of 0.6. So this is a member of the blue group, and this is clearly a member of the green group, and it happens very discretely somewhere at this region in the pi helix. This 218 is very important because you'll notice on the y-axis in the reefer, this is log L2. You can see there's 6th order of magnitude range that you can get from Y to A and mutations of this residue. This is the most sensitive residue we've found in the receptor so far. This is a very important region. So there's a very discrete break in Phi values. Now these, if you notice above the 217, there's this conserved R209 in the linker region, which helps hold the thing together. And there are two red residues here, and that's a low Phi of 0.3. And you may ask, well, what is this low Phi residue doing in this high Phi region of 0.8? Well, we don't know, but it turns out in structures, these residues are in contact with lipids. And we are speculating that there is a very late perturbation of the lipids, and that perhaps that these residues are blue, but they get a secondary energy bump at the end of gating after or simultaneous with the water compartments fusing. There's a energy perturbation that adds to the one early in gating to give you an average of 0.3. This is one of the complexities of Phi value analysis. So some of the scatter that you see in Phi values arise from complicated things like Hammond effects and secondary perturbations. But notice that the proline that causes this formation of the pi helix is just another member of the green group. It doesn't have anything special going on with it. So this is the end of the second part of the 4th chapter, which is what we learn from the Phi distribution, or how we interpret the Phi distributions. There are 5 intermediate transitions, 4 intermediate states. We learned the sequence: hold, twist, tilt, rip, and pop. And of course, in channel closing, it's the reverse sequence: pop, rip, tilt, twist, and hold, going backwards. We know the shape of the barrier roof and the position of the transition state within that region. We can get a dynamic map of energy change within the gating conformational cascade. And we can locate important critical structures that lie at Phi boundaries, for example. And in the next chapter, the final chapter, we're going to try to synthesize all this into a dynamic picture of a zipper for the conformational change.